What evidence that?
Please do not drop your name in the chat room. Please do not drop anything in the chat room. I don't need the others in the chat room, please.
I was there 15,000. I'm looking at get ready. I'm going.
Those of you that you are dropping your name, just wait to be removed. I'm going to remove all of you one by one if I start this class. And I will put you in the waiting room to be able to join the class again. I said, don't drop your name in the chat room. If you do, I'm going to remove you from this platform. Go and join on YouTube. All right. Good morning, everybody, and welcome to today's class. I think I will just make this. Sadly, some of you cannot follow instructions. I don't know why. Right? I don't know the what was it called? Why you can't do but dropping your name in. I've told you several times. I don't need your attendance here. Some of you even join, and some of you are on the road. Some of you are in your house, not even paying attention to the lecture. You only want to drop your names as if dropping your name you've passed the course automatically. No, please. So I've told you several times, I don't need your name in the chat box or whatever whenever you join. Just pay attention. And if you don't pay attention, you're on your own. Joining Zoom does not give any advantage over those that are on YouTube. You need to pay attention. It's not just dropping your name, then you're going to be doing whatever you are doing, thinking that nobody sees you. You are deceiving yourself. So please let this be the last time you drop your name when we are having this class, or else I will compile that list and I'm going to deal with you. That list that you are sending, I will compile it and I'm going to use it to punish you. All right. Last time we started the biomolecules and the central dogma of life. We looked at the biological molecules, the molecules of life that are important. We look at the four classes that we have: the carbohydrates, the lipids, the nucleic acids, and proteins. And we talked about their monomers, stated some functions, and we dealt with mentioned some structures of them, what they have in common. For protein, for instance, we said the monomers are the amino acids, and we looked at the structure of amino acids having a central carbon that have four major things attached to it, of which three are most important. The R side chain that determines the type of amino acid that we are going to have, whether it's going to be acidic, it's going to be aromatic, or what have you. Then we have the amino group and we have the carboxylic acid group that get attached to that alpha carbon. Then we said carbohydrates, they serve as the source of energy, and their monosaccharides are the major monomers that are joined together by glycosidic bond. And we said nucleic acids have their, okay, we've not gotten to nucleic acid. Then we talked about lipids being not a polymer, right? Because it doesn't have monomers. And then we said it's majorly triglycerides and fatty acids, three molecules or three units of the fatty acids and glycerol. Then we said we have phospholipid, in which one of the hydroxyl group is replaced with phosphate groups, and we said these form the basis or the major lipids that we have in cell membrane, your cell membrane or the plasma membrane. And we said the phospholipid is a bilayer, right, that separates the cell from the outside environment. And we said we have the phospholipid as the head, that is hydrophilic or water loving, and the tail, that is hydrophobic or water hating. Then we talked about what's it called? Okay, so many things. So let's today, I think we start at slide 20 or 21. So today we are going to go, we are continuing the lecture. Okay, we've looked at, we've dealt with all of these ones. We've dealt with amino acids. Okay, we also look at structures of amino acids, that the linear arrangement of this amino acid sequence are the primary structure of protein. Then the polypeptide folds into alpha helix structure in secondary structure. Then we have a 3D structure in tertiary, and two or more folded tertiary structure forms the quaternary structure of protein. So today we are going to continue with nucleic acids. Now, you've all heard of DNA before, right? DNA testing, paternity testing, and what have you. So these nucleic acids are what we refer to as DNA. Right? So the nucleic acids are molecules that store information for cellular growth and reproduction. So they are the blueprint of life. The nucleic acids are the blueprint of life. They hold the information for traits, for cellular activities, and what have you. right? And there are two major types. We have the DNA, the deoxyribonucleic acid, and the RNA, ribonucleic acid, right? Then the monomers are nucleotides. The elements are carbon, hydrogen, nitrogen, oxygen, and phosphorus. So last time we talked about atoms being the basis of biomolecules. These atoms will combine to form molecules, and these larger molecules will combine, those molecules will combine to form a larger molecule known as polymers or macromolecules. So the atoms of nucleic acid that you can see in nucleic acids are oxygen, as we have it here. We have oxygen here, we have phosphorus here, we have carbon, we have hydrogen, and we have nitrogen, right? So these are major elements that form nucleotides, and these elements or atoms are held together by covalent bonds. But when nucleotides join together, we have phosphodiester bond. So a nucleotide is made up of three major parts or components. They are the nitrogenous base, 1, a pentose sugar, 2, and the phosphate group, as we have on the screen. So let's take it all over again. We said nucleic acids are a kind of polymers. There are two types: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Then we said these polymers, the nucleic acids, have monomers known as nucleotides. Then nucleotides have subunits. What makes them to be called a nucleotide? Right? So there are three things that come together to form a nucleotide, and those three things are: number 1, a nitrogenous base; number 2, a pentose sugar. Pentose sugar means a sugar with 5 carbons, 5 carbon atoms, pentose sugar, right? Then phosphate group. So these are the three major components of a nucleotide. Then we have nucleoside, and that will be an assignment number 1 for you today. What is a nucleoside? How is nucleoside different from nucleotide? Nucleoside, just replace the T in nucleotide with an S. You have nucleoside. So what is a nucleotide? Nucleoside. How is it different from a nucleotide? Okay, so that is that for structure of a nucleotide. Then this is how they combine to form a nucleotide. The three components, right? We have a pentose sugar, right? So the pentose sugar, the carbon is labeled 1 to 5. 1, 2, 3, 4, 5. That you can, the 1, 2, 3, 4, 5 that you can see, those are the 5 carbon atoms of the sugar. Pent, from your organic chemistry, pentane, your 5 carbon hydrocarbon. Okay, pentose, pent is a pentagon, 5, whatever. Whenever you have pent, is actually 5. So 5 carbon sugar, pentose, a pentose sugar. Now we said nucleotide has 3 components, and they combine in a way to give the nucleotide. So at carbon 1, we have the nitrogenous base. So the nitrogenous base will always attach to carbon 1 of the sugar, as we have it here. This is the carbon 1 of the sugar. That is where you have the nitrogenous base. That is depicted with N base, nitrogenous base. We are going to look at nitrogenous bases later. Then at carbon 5, you have the phosphate group. The orientation is always like that, all things being equal, right? So you have the carbon 1 of the sugar carrying the nitrogenous base, and carbon 5 having a phosphate group attached to it. Now, if you observe the carbon of the sugar that we labeled 1 to 5, is that there is a prime on those numbers: 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. The reason why we put prime on those numbers is to distinguish them from the carbon of the nitrogenous bases, because the nitrogenous bases also have sugar. They are made of carbon atoms. They also have carbon atoms in their structure. So for us to know, okay, this carbon is for the sugar and this carbon is for the nitrogenous base. So we put a prime on the carbon of the pentose sugars. So that is why you can see you are seeing 1 prime, 2 prime in red, 3 prime, 4 prime, and 5 prime. 5 prime. So that is the major difference between carbon of nitrogenous bases and carbon of our pentose sugar. Now these are nitrogenous bases. Nitrogenous bases are of 2 types. Again, we have the purines and the pyrimidines. Purines are double ring bases, whereas pyrimidines are single ring bases. As you can see here on the screen, we have 2 rings, 1, 2. But for our pyrimidines, they are just 1 ring. So we call them 2 ring bases for purines and 3 ring bases for pyrimidines. Examples of purines are adenine A and guanine G. Examples of pyrimidines are cytosine, thymine, and uracil. Now, we said DNA and RNA. Here, let me tell you one of the differences between the 2 types of nucleic acids. Don't forget, DNA and RNA are types of nucleic acids, and we have them in organisms. Some organisms have DNA, like humans, for instance, while some lower organism prokaryotes have RNA as their genetic material. For instance, your HIV virus, they are RNA viruses, right? So we have some viruses as their genetic material, they don't have DNA. There are some organisms like humans that have DNA. Okay, now one of the differences in DNA and RNA is that the bases that you can find in RNA are adenine, guanine as the purines. Then for the pyrimidines, you can only have 2 types out of the 3 that we have. We have cytosine and thymine. So we don't have thymine in RNA and we don't have uracil in DNA. I hope that is clear. We have purines and pyrimidines as our examples of broad examples of nitrogenous bases. So under the purines we have A and G. Purines PAG, pyrimidines cytosine, thymine in DNA, and uracil in RNA. So RNA does not have thymine, DNA does not have uracil. So that is one of the differences between DNA and RNA. Let's move on. Sorry. Then we also have this slide explains our pentose sugar. So we are looking at those components of nucleotides. We have looked at pentose, we have looked at nucleotides, the DNA, the RNA, we have looked at nitrogenous bases, we have looked at purines, we have looked at pyrimidines. Their examples: pyrimidines are adenine and thymine, A and T. Pyrimidines are cytosine, thymine, and uracil. Now, we said these nucleotides have pentose sugars, but the sugars are different in DNA and RNA. Another example here. Now, we said there are 2 related pentose sugars. RNA contains ribose sugar and DNA contains deoxyribose sugar. So the pentose sugar can either be ribose or deoxyribose. In RNA it is ribose, in DNA it is deoxyribose. And I said this earlier, that sugars have their carbon atoms primed to distinguish them from the carbon of the nitrogenous bases. Now, let's look at the pentose sugar in DNA and in RNA. Now, this is a pentose sugar that we see in yellow before us. And like I said earlier, their carbon atoms are numbered 1 to 5 with prime. And I said on carbon 1, it is always the nitrogenous bases. But on carbon 5, you have our phosphate group. Now, the difference between DNA and RNA in terms of sugar, let's look at it. If you look at carbon 2 of RNA, for instance, carbon 2 prime of RNA, we see that there is an OH that is attached to that carbon 2. If you look at carbon 2 of DNA, there is ordinary H. The oxygen atom of that hydroxyl group is lost. no oxygen is bonded to this carbon, as you can see there. So that's why we said the oxy, there's a removal of an oxygen atom from the carbon 2 of DNA. So that's what it says the oxyribonucleic acid, that is, there's a removal of oxygen at some place in that DNA. But in ribose sugar, it is OH. So the 2 is there. Then if you look at carbon 3, both of them have OH. Please pay attention to that OH of carbon 3. We are going to refer to it later. Right? OH is always attached to carbon 3 in both. But in RNA, it is OH. In DNA, it is H, that is in carbon or at carbon 2. So that is another difference between DNA and RNA. Assignment number 2 for today. Mention other differences between DNA and RNA. I've given you 2 now. We have looked at the different types of nitrogenous bases. We have looked at the differences in terms of their pentose, their sugar. So what other differences exist between DNA and RNA? I need you to tell me in your notes, not in the comment section, please. Okay. Then pairing of nucleotides. We said nucleic acids are polymers of nucleotides. We have nucleotides come together to form polymers known as nucleotides. So how do these nucleotides combine? Don't forget, we said when monomers come together, there's a form of a reaction that takes place, right? Now, let's look at how they pair up. Conventionally, the purines will always pair with pyrimidines, and pyrimidines will pair with purines. Don't forget, we said that our purines are adenine and guanine, and our pyrimidines are cytosine and thymine. Now we are looking at DNA now. Let's leave uracil, which we see only in RNA. So the 4 bases that you can see in DNA are adenine, cytosine, guanine, and thymine. Let's face these ones. The other one, uracil, is seen in RNA. When we get to where we are going to use that, we will recollect it. Now, we said that purines will pair with pyrimidines, and our 2 purines are adenine and guanine. By convention, A pairs with a T, G pairs with C, and vice versa. So A will pair with a T will pair with A, or C pairing with G, or G pairing with C. Now, when the nitrogenous bases pair, when they pair up, when A pairs with T, don't forget A is a purine that binds with T, a pyrimidine, right? So when they pair up, there is a type of bond known as hydrogen bond that hold them together, and it is 2 hydrogen bonds that we have between adenine and cytosine and thymine. Then between C and G, we have 3 hydrogen bonds. Now, which of them will be more stable? Is it the one with 2 bonds or the one with a double bond? If you are to break that bond, which one will require more energy? Okay. Now, we said adenine pairs with thymine with 2 hydrogen bonds, and guanine pairs with cytosine with 3 hydrogen bonds, and vice versa. Whether A pairing with T or T pairing with A, you have 2 hydrogen bonds. Whether guanine pairing with cytosine or cytosine pairing with guanine, you have 3 hydrogen bonds. Now, let me tell you one beautiful thing about DNA. DNA is helical in shape, and it runs, in parallel. So you can have a stretch of DNA that looks like this. So if on this segment of DNA, we said DNA is a double-stranded, 1 strand and 2 strand, the second strand. So if on this strand, for instance, you have these bases, A, T, C, C. So for the other strand, you are going to have the other nitrogenous bases pairing up. So for A, so let's form the other strand for this one. We have a T, yes or yes? Oh, no, sorry, let's use, okay, this one is also clear. You have a T. Then for our T on one strand, you have an A on the other strand. For our C on this strand, you have a G. Then for another C, you have a G. Then if you have G here, for instance, if you have G here, then you are going to have a C on the other strand. So that is how the bases pair up in DNA. So that is why you can have this side here now and this side. So this side we have T, T, G, G, A. Whereas on this side we have A, C, G, T. So that is how they pair up. They run in a parallel. Then look at what I said earlier about carbon 3. Okay, I think there is a better slide that explains what I want to say just now. So when we get there, we will look at it. Okay, now this is formation of the phosphodiester bond. The other time I said you pay attention to that hydroxyl group of that carbon 3. You can see the hydroxyl group highlighted in yellow there. Now, when the nitrogenous bases want to pair up, for instance, this is one base, maybe this is the nitrogenous base, this is the pentose sugar, this is the phosphate group, so this is a nucleotide. That is a complete nucleotide. Now, when another nucleotide wants to pair up with this one, this is like a growing chain. This is like a growing chain. This is one nucleotide, this is another nucleotide. This one is single ring, this one is double ring. I'm sure you know which one is purine, which one is pyrimidine between the two now. Now, so when they want to pair up, they want to form a long chain of nucleotides, right? They are going like this. So this one is the first one here, for instance. This is the first one. Then the second nucleotide wants to attach to the first nucleotide so that they can form a long chain of nucleotides known as nucleic acids. Now, that carbon 3, the OH needs to be there because that is where the OH of the phosphate group will attach to form the phosphodiester bond that we say forms between nucleic acid. We said when nucleic acids join, there's a phosphodiester bond that is formed, right? Now, this is the phosphodiester bond here. This is the phosphodiester bond here. So the OH needs to be available at that position 3. That is where the carbon atom, that is where the hydroxyl group of the phosphate group, what we have here. So there is going to be OH plus OH, and there will be a removal of H2O. Then the remaining O will be used to pair up with the other nitrogenous bases. So that is how their own condensation or dehydration reaction takes place. Don't forget, in our last class, we mentioned of how polymers are made and how we can break them down by condensation or dehydration or by hydrolysis. We make them by dehydration or condensation. We break them by hydrolysis. So this is how our one molecule of water is lost between one nucleotide and the other one that is forming a growing chain. Another thing I need to pay attention to here is the orientation of the nucleotides. Now, this is our nucleotide, this is the side of interest. When you look at this nucleotide and this nucleotide, when they form in this way, when they form a growing nucleotide like this is the product that is formed when this guy attaches with this guy. So we have this guy here. Now, here we have what we called the orientation of these nucleic acids that are formed. Look at this. we have the 5 prime that is exposed at this end. Then here we have the 3 prime that is exposed, right? So we said this strand runs from 5 prime to 3 prime. I'm sure you are getting it. We're looking at, because you said DNA is made up of 2 strands, like I explained earlier, and the strands run anti-parallel. That is, one runs in this direction, the other one runs in the other direction. So this one, I want to tell you how it will form when you hear 5 prime to 3 prime or 3 prime to 5 prime later in the course of this class. I want to show you what we mean by 5 prime and how they are formed. Don't forget we said earlier that our carbon sugar, the carbon of the sugar is primed to disintegrate from the carbon of the phosphodiester of the nucleic acids of the nitrogenous bases. Now, so when that happens, there are ends that are exposed when we have a phosphodiester bond. When those nucleotides come together to form long chains of a nucleotide known as nucleic acids. This end that carry the phosphate group is exposed from this end, and that is the 5 prime. Don't forget we said the phosphate group will always attach to the 5 prime. Then the OH, the original OH that you have here from the second one, it is this one. So the 3 prime is exposed, so that another nucleotide that is coming with its OH, which is PO with phosphate group, can attach to this free OH. So that is why this end is always exposed. The 3 prime carbon is always exposed. We're still going to mention 3 prime OH later in the course of this lecture. Okay, so this guy runs from 5 prime to 3 prime, and the other strand of nucleic acids that we are going to have will run anti-parallel from 3 prime to 5 prime. So that is the orientation of DNA. What we mean by 5 prime, 3 prime, and 3 prime, 5 prime. Okay. So structure of DNA. Several scientists elucidated the structure of DNA, but people that were recognized for that structure were Francis Crick and Watson and Crick in 1953 determined the structure of DNA, and they won the Nobel Prize for that. And they said that the DNA is a double helix that is made up of sugar phosphate backbone on the side with bases on the inside. So what that means is that you have heard of sugar, you've heard of that is ribose sugar, you've heard of phosphates, and you've heard of bases before, the 3 components of a nucleotide. So what they mean that it's a double helix, like your ladder, right? The DNA is a form of a ladder, double helix, one strand and the other strand of a ladder. So the backbone, when you look at a ladder, this is a ladder, for instance, this is a ladder, right? So the rungs of the ladder that you put your leg as you are climbing is like the nitrogenous bases, they are inside. Then where they stand by the side are the phosphates, the phosphate and sugar backbone. So the backbone is the phosphate and the sugar, where stands on the floor. Then the rungs of the ladder are the nitrogenous bases. All right, I'm just trying to liken the DNA to what you can comprehend or get familiar with. Now, they say that it's double helix, right, and helical, right? What they mean by that is that it is like a spiral staircase. If you have been to Faculty of Life Sciences before and used the spiral staircase, you see that is the structure of a DNA. Then like a spiral staircase, wound around, the 2 helix are wound around each other in form of a helix. And before them, there are some other scientists that have worked on the structure of DNA. We have the Rosalind Franklin. We have so many other scientists that I'm going to give you as an assignment. Okay, we have, like I said earlier, we have Rosalind Franklin's, we have Chargaff, and there's some other scientists that work on the structure of DNA. So this guy, Rosalind Franklin, X-ray images suggest that DNA was a double helix of even width. So this is Rosalind Franklin, and she postulated this, right, that when observed under the X-ray, the DNA is like this, but it was not able to give actual structure of the DNA. So it was Francis Watson and Crick that actually gave us the structure, and they took the credit. But they did not work alone. Their research built on studies or works of other people before them, like Franklin, like Chargaff, and what have you. Then Chargaff, excuse me, Erwin Chargaff was one of the scientists that contributed to DNA structure. And what he says on his own in his own contribution was that the amount of our nucleotides, they are relatively equal in DNA. And what he said, that the amount of adenine in a living organism is equal to the amount of thymine. And if you look at it, if we have A and T pairing up, so if you have 100 A in an organism, definitely you are not going to have 200 T or 101 T, for instance, because A will always pair with T and C will always pair with T. So the amount of adenine and thymine are supposed to be equal, and the amount of cytosine and the guanine are expected to be equal. And this is like, so he said, Chargaff will say that A equals T and C equals G. So we expect equal amount of adenine and thymine and equal amount of guanine and cytosine, and that was credited to Erwin Chargaff. That was his own contribution to DNA when we talk about the amount of DNA. Then let's quickly look at this example. We said, based on Chargaff rules for the base composition of double helical DNA, if a sample of DNA contains 36% thymine, what are the percentages of adenine, cytosine, and guanine in the DNA? I think I should give you some minutes to do this and give me your answer in the chat box. So attempt the question on the board and give me your answer. The assignment, when and where are we to submit the assignment? Do it in your notes and let it form part of your notes. I don't collect assignments, but do it inside your notes and let it form part of your notes. I'm sure you are not just attending this lecture for fun. As you are joining the lecture, take notes and study, right? So that is why we are sending the recording of the lectures to you, so that you can always fall back on it to read. So if we have 36% adenine, what's in an organism, for instance, so what is going to be the percentage of the other 4 molecules, or what's it called, nitrogenous bases? So let's see answers in the chat room. What do you have for your T? Just give a list. A equals this, C equals this, G equals this. Okay, we have answers, we have answers, we have answers, we have answers. Okay, we have seen different answers. We are seeing different answers. We are seeing different answers. We are seeing different answers. Okay, let me see whether I can connect with my guys on YouTube and see how they are faring. Enjoying the lecture as well. Good morning, you. Okay, I can see some answers on YouTube. I'm glad to know that you are also contributing. Okay, we have 36%, A 36%, 9%. Different answers on YouTube as well. C equals 14, A equals 36, C equals 14 from this King 5R. Then Teniola, okay, that is said C equals 14%. Oluwa Oluwaseyi Feyi Shayo. What is this name? Okay, a beautiful name. A equals 36, C equals 14, G equals 14. Okay, then Foxy Frosh. A equals 36, C equals 14, G equals 14. Yeah. Good attempt, good attempt, good attempt. So let's quickly explain, because I saw some wrong answers as well. So let's quickly explain the logic behind our A equals 6, G equals. So Chargaff said that the amount of A equals the amount of T, and the question says that T is 36%. Just slot, that means A is also 36%, all things being equal, right? And we're looking at percentage, 100. So if we have one, A is 36 and T is 36. 36 plus 36 is 72. If we take away 72 from 100, we have 28. If we divide 28 between C and G, we have 14 each, because they must be the same. So definitely our A equals 36, T equals 36, C equals 14%, and G equals 14%. Okay. So structure of the DNA, like I said earlier, the DNA is a double helix with a spiral case, and we can see like a radar, like with phosphates, sugar backbone, and the bases forming the rungs. And we can see there the G, the C, the A, and the T, how they pair up. So these are, okay, I even have examples of DNA, difference between DNA and RNA, and I'll give you as assignment. Okay. RNA, ribonucleic acid, how is it different from DNA? RNA is a messenger that allows instruction of DNA to be delivered to the rest of the cell. We are going to look at that better and get to central dogma, how the information encoded in the DNA is read and translated by the RNA. So we are going to explain that bullet point as in terms of when we get to central dogma. So RNA is different from DNA in that, one, the sugar in RNA is ribose, the one in DNA is just ribose. RNA is single-stranded, DNA is double-stranded, like we elucidated and explained earlier. RNA is found inside and outside of the nucleus, while DNA is only exclusively in the nucleus. Apart from the nucleus, we can find our DNA in chloroplasts and mitochondria. These are the other organelles that have their own DNA. The chloroplast, the nucleus, and the mitochondria, they have their own DNA. Then the bases in DNA are GCAT, the bases in RNA are GCAU, T in DNA, U in RNA. Okay, the central dogma of life. So we are getting to the end of the story or of the lecture. Okay, we said the central dogma holds the genetic information expressed in a specific order from DNA to RNA. Now, let's look at this scenario. The information, excuse me, the information that the cell needs. We said, don't forget, we said that cell is the basic structural and functional unit of life. There is no life apart from the life of cell. So if you don't have cell, you don't have life. So you operate at the cellular level. Your skin color, everything about you is about cell, is about your DNA. So there is what we call protein. There is DNA that codes for that protein. So the information is encoded in the DNA, and it is only expressed when it is written in form of a protein. So the central dogma simply talks about the flow of information from DNA to RNA to protein. So the information in the DNA, the cell does not understand what a DNA is. The cell only works with protein. Protein, don't forget I said, what is it called? All enzymes are proteins, or what do we? Yes, all enzymes are proteins, but not all proteins are enzymes. So those enzymes, you know what they do, you know what proteins are now. So proteins are like effector molecules. They are the ones that will go, okay, go and do this, go and do that, change this color to black, make this one an albino, do this with some enzymes. Okay, so the protein is what the cell, is the language that the cell understands. Protein is the language of the cell that the cell understands. But the information that the cell will use is in DNA. So you need something that will translate that information to what the cell understands. The information encoded in the DNA needs to be translated to what the cell understands. Just like maybe for instance, you are a Yoruba guy, right? Then and you want to communicate, an Hausa man needs to pass an information to you. The Hausa man does not understand Yoruba. Then you, the Yoruba, you don't understand Hausa. So you need an Igbo, right? Or Igbo that we interpret, that understands both Hausa and Yoruba. Are you getting it now? There's an Hausa that wants to pass information to Yoruba. So you have Yoruba at the receiving end. Then the Hausa man does not understand Yoruba. The Yoruba man does not understand Igbo, does not understand Hausa. So you need an Igbo that will stay in the middle as a middleman, as your interpreter, as your messenger. That when the information is passed from the Hausa guy to the Igbo, then the Igbo will say, this is what he's telling you to do. He said, run. Okay, you two will now pick your race and start running. That is simply what the central dogma is all about. So the central dogma is the information encoded in the DNA translating to protein. Then how does that go? The information flows from DNA to RNA, then to protein. So your DNA is the Hausa guy, your RNA is the Igbo, your Yoruba is the protein. That is simply what the central dogma is all about. The flow of information from DNA to RNA to protein. I think that is. I have some things that we circled in this structure. So we are going to be looking at the process of replication, transcription, and translation. So replication simply means DNA making more of its copy. DNA replication, and this occurs during the S phase of your cell cycle. If you remember your cell cycle very well, secondary school biology, you have the cell cycle at the stage of growth that the cell undergoes before it divides, right? And the cell cycle is divided into 3. You have the G1 phase, the S phase, and the G2 phase before the cell enters mitosis, right? So the S phase, the synthesis phase, S for synthesis, is where this DNA is duplicated before it divides, so that you can have more copies of DNA in the daughter cells. When you get to cell division, you will learn more on that. Now, so we have DNA replication. What that simply means that DNA replication is a process whereby more copies of DNA are made. Simple, DNA replication. Then we have transcription. Transcription is the process whereby RNA is made from DNA. So the information in the DNA is transcribed to RNA. The information in the Hausa guy is passed to the Igbo guy. Are you getting it now? Then we have mRNA to protein, known as a translation. So the process whereby the message in the mRNA is translated to protein, known as a translation. Is right. So, or protein synthesis, and our protein synthesis takes place on ribosomes. Dr. Otu will come in and take you cell structure and function. We look at certain organelles in the cell and what they do. And from secondary school biology, you will have heard of ribosomes before for protein synthesis. Now, the process of that protein synthesis now is what we are going to look at here. How do we make the proteins? How do ribosomes make proteins? Right? Okay. So that is basically central dogma. Then we can have RNA back to DNA. That's another assignment for you today. What is the process of RNA conversion to DNA? What is it called? Let me take. You say we can make DNA from RNA. What is that process called? And what is the name of the DNA that is formed? What is it called? Okay, so this is basically the central dogma of life, right? So we can have our DNA to DNA, known as replication, then DNA to RNA, known as transcription, and RNA to protein, known as a translation. Okay, let's take these processes one by one. Replication, making of more DNA. Transcription, RNA synthesis from DNA. Then translation, protein synthesis from the RNA that we make. And the type of the RNA that we are referring to here is the messenger RNA. As I mentioned, number 4 for today. What are the other types of RNA that we have? Okay, there are many of them. So let me not confuse so that I won't go and be looking for what you don't need. Other types that we have are transfer RNA and ribosomal RNA. So the assignment I will give you there from there is that what are their functions? What do they do? What is the function of ribosomal RNA, rRNA, and transfer RNA, tRNA? We have other types of RNA. We have long coding RNA. We have so many types of RNA. But for your level, we are looking at those three. The messenger RNA, tRNA, transfer RNA, and rRNA, small letter r RNA, small letter t RNA for transfer, and small letter mRNA, messenger RNA. So the one that you make here in this project that we are going to be familiar with is the messenger RNA. And we are going to make reference to the other types of RNA later in the course of this lecture. The ribosomal RNA and the transfer RNA. So before we get there, if you don't get there today, go and read up transfer RNA and ribosomal RNA and what they do, so that when I get there, I will just flip through like I have taught you. All right. So let's move on. Let's look at the process of DNA replication. So DNA replication is the biological process by which a cell makes an exact copy of its DNA, producing two identical molecules from one original molecule, usually before cell division. Like I explained earlier, DNA replication takes place before cell division. That is at the cell cycle stage, right? So that is the S phase of the cell cycle, and more copies of DNA are made in the process as DNA replication or duplication or synthesis. Now, the process of DNA is semi-conservative process, meaning that when you have two strands of DNA, for instance, you have these strands of DNA. So by semi-conservative, we mean that the new DNA strand that you will form, don't forget we said we are making more DNA copies, yes or yes, in DNA replication. Now, when we have this originally, the new DNA that we are forming, we have one original or parental strand and the other original or parental strand. So from this original DNA, two strands, we have made two copies. So we have this new copy, and another new copy. So we have made more copies of DNA, and that is what we mean by a semi-conservative model of DNA replication. That is, there is one original or parental strand and the one newly synthesized strand in the new DNA that you form. It's like doubling the DNA, the number of DNA. So when you have one DNA, when it replicates, it becomes two. So the two new DNA strands, the new daughter DNA that you have now, one of the strands, don't forget that DNA is double-stranded, right? One of the strands, this is what I am explaining again. This is a strand of DNA. This is one molecule of DNA, for instance. When these guys replicate, it gives us two, right? So the two is that one of them, this is the first daughter DNA, and the other daughter DNA. So by semi-conservative, we mean that the two cells that are formed, the two DNA that are formed, this is one, this is two. One of the two strands is from the parents, right? From this guy, and the other one is from this guy. So that is semi-conservative. Scientists propose different models, but the accepted one is the semi-conservative model, whereby the new strand has one original strand and the newly synthesized strand. How this is done, we are going to look at it. Okay, so these are the three general models of DNA replication that was postulated before we settled for semi-conservative. So let's quickly look at the three models. We are running against time now. Now, the semi-conservative model, like I said earlier, each daughter strand has one parental and one new strand. For the conservative model, the two parental strands stay together. Then for the dispersive model, DNA is fragmented and both new and old coexist in the same strand, and their examples are depicted illustratively, illustrated here. Now look at this semi-conservative. We said the new strands, right? One is parental, the other one is new. But for the conservative is like, no, the parental strands are conserved. You have them staying together. That's why you can have, I don't know color, that's not color green, I'm sure, and it's not color blue, I'm also sure, right? I don't know this color. So whatever this, so let's, what is this color? You people that know color very well, what is this color? I don't know. I'm a bushman. All right. So that is our color. They said that the, let's call it the parental DNA. The parental, okay, maybe somebody might even drop it in the chat room. I trust my students. They are brilliant. They are sharp. Somebody should help us drop that color in the chat room. Those of you that's no color. Hey, Zion. I said that. You people, Zion. Okay, let's take. I think the Zions have. It's okay. It's okay. Turkish Zion. Don't worry. Turquoise. Is it turquoise they call it? But don't worry. Can pronounce Zion better. I will go with Zion. I don't want to remove my teeth this morning. Thank you. Okay, the Zion color and the red color. So the Zion is the parental color, right? So the semi-conservative is saying that when we are forming our new daughter DNA, right, that the Zion, which is the parental strand, will stay apart. They will separate, they will open up, and they will form the basis for the new one. So there is one parental strand and there is a new strand indicated as red. But the conservative says no. The parental strands are conserved. They stay together, whereas the new cells, the new strands are also together. Whereas the dispersive said no, that is not how it is like. Oh, it is that everything is fragmented. The new strands is fragmented, the parental strands is fragmented, and they now form together old new, old new, old new, old new, parental new, parental new, parental new, parental new. But based on the what was it called? The research of these scientists, Meselson-Stahl, they were able to confirm that the actual model for DNA replication is the semi-conservative, and this is their research that they did, and they found out that, and what they did was simply they cultured bacterium in a medium containing heavy nitrogen, right? They now transfer it into a medium containing a light nitrogen, and we have isotopes, if you remember correctly from secondary school chemistry. So we have 14 nitrogen and we have 15 isotope nitrogen. So what they did was to first culture the bacterium in a 15 isotope 15 nitrogen, known as heavy nitrogen. Then they let it to replicate, so they have more copies of it. They now transfer it to the light nitrogen, so they let it to replicate again at the centrifuge, and they were able to confirm that, okay, the new strand in the first generation after we have transferred it into, after 20 minutes, is that this, what we are seeing here, is more like the semi-conservative model, because they are seeing DNA that have the new strands and the old strands. And you may want to read up on that experiment by Meselson-Stahl. So describe the Meselson experiment, and you may want to answer those questions. So if you read that, if you read the assignment, if you studied the experiment, you will know what it is all about, and you'll be able to understand better. It's just an experiment to confirm the models for DNA replication, and that experiment confirmed that it was a semi-conservative. So before we go deep into DNA replication, okay, I think we should stop here for today. So on Friday, by God's grace, we will go deep into DNA replication, and we will end the class by God's grace. So maybe we have 1 or 2 questions. The class is over now. If you don't have any questions, you can go and join your next class if you have one. And if you have any question, you can raise your hand in the chat room and you'll be allowed to, I said in the chat room, you can raise your hand and you'll be allowed to unmute yourself and you will ask your question. Can someone list all the assignments he gave? If you paid attention, you will take the assignments as I was giving them now. Okay, maybe on your class groups. Me, I don't have them. I just give as they come, as the Spirit leads. So I don't have them written anywhere. Okay, if there is okay, maybe before the next class, you guys should read up on DNA replication, translation, and transcription, so that we will just run over it. So these are the enzymes that we have in DNA replication. So just read up DNA replication, translation, and transcription in our next class. I don't think anybody is raising their hands here. Okay, Daniel Damilola Alabi is raising his hand. So you can unmute yourself, Mr. Daniel, and
Hello, sir.
yeah, good
Good
morning.
morning, sir. Sir, please, can you explain on this nitrogenous base?
Okay, the nitrogenous basis. Okay.
Yes, sir.
Okay. Okay, let's take Chinecherem, then we answer the two together. You can lower your hands, Alabi. Thank you. Chinecherem, please unmute yourself and let's hear your question, please.
Yeah, please, for the
You said?
for that monomers,
The monomers of what exactly?
the monomers
Monomers
of
you said?
the nucleic acid.
Monomers of nucleic acids. Okay. Okay.
Yes, sir.
Now, we said that, please, you can mute yourself back if you have your question. So we said nucleic acids, their monomers are nucleotides, right? And a nucleotide has 3 components. There are 3 things that come together to form a nucleotide. And those 3 things are nitrogenous bases, pentose sugar, and those sugars differ in DNA and RNA, and a phosphate group, right? Nitrogenous base, pentose sugar, and phosphate group. Now, the sugar in DNA is deoxyribose because there is a loss of oxygen atom at carbon 2. The sugar in RNA has the oxygen. So for carbon 2 of RNA, you have OH, hydroxy group. For carbon 2 of DNA, you have an hydrogen or a hydrogen. Now, that's basically the nucleotides. And we said there's a phosphodiester bond that forms between one nucleotide and the next. And this is how the phosphodiester bonds are formed, right? By the incoming nucleotide attached to the 3' hydroxy group of the existing nucleotide, right? Then the first question was on nitrogenous bases. So the nitrogenous bases are a component of a nucleotide, like we said earlier. We said there are 2 basic types, the pyrimidines and the purines. The pyrimidines are adenine and guanine, and the pyrimidines are cytosine, thymine in DNA, and uracil in RNA. Now, when these are pairing up, we said adenine will pair with thymine with 2 bonds, and cytosine with guanine with triple hydrogen bonds. And that is basically what we said on nitrogenous bases. And we said the purines are double ring. They have 2 rings, whereas the pyrimidines are single ring. I think that is basically what we say on this, what's it called? On the nitrogenous basis. On nitrogenous basis. Okay, think we have come to the end of today's class. You can join another class if you have one. Do have a wonderful day and stay out of trouble. And okay, if you are still there, we are going to release the lab grouping today by God's grace. By the time I will be done with it before by noon, as it is, and whenever it's ready, I'm sure it will be out today. So we are releasing the first set or batch 1 today. So by God's grace, next week we are going to release the last one. So the people that we see their name today are those ones that have their practical on Monday. And these are people in the department of biochemistry, cell biology and genetics, chemistry, marine sciences, medicine and surgery, microbiology, nursing science, pharmacology, pharmacy, physics, and psychology. If I mention your department, biochemistry, cell biology and genetics, chemistry, marine science, medicine and surgery, microbiology, nursing science, pharmacology, pharmacy, physics and psychology. If you hear your department and you have registered for practical, I intentionally left that thing till yesterday instead of closing on Monday. Some people say registered as late as 5 p.m. yesterday. So if you registered already and you are in those departments I called, your grouping will be out today and you have practical next week, Monday, January 12th, by God's grace. So if you have not gotten your manual, go and get your manual. And I plan to have briefing on Saturday, practical briefing on Saturday. I will work towards it. The time I can't say for now. I will look at my schedule for Saturday so that I will tell you what we expect of you and the do's and don'ts of our practical. Get your manual. If you come with an old manual, you will come back and do the course next year. That means you are not ready for the practical this year. So get your manual. Don't come with your brother's manual from last year. Get your lab coats. Dress properly. You wear closed-toe shoes. We don't allow Crocs. We don't allow sandal. We don't allow slippers. If your leg is paining you, come with a medical report that says that that leg is actually paining you. That's why you put plaster of Paris, POP, not because you don't want to wear your shoe. So please dress properly, get your lab manual, and know your time. Don't come when it is not your time. You miss your practical, you miss it forever. So please follow these instructions. I seriously think I will be able to have that session with you so that we can tell you what we expect of you, so that we have little cases in the lab or complaints, because if you misbehave, you can be sent out of the lab and you have lost the mark for that day. So please get your manual and write, most especially or importantly, write your name on the first page of your manual. Okay, second page, when you open it, there's a place for name, your matric number, your department, your level, your group and table number. You will have your group and table number today. List them out on your manual, right? Fill it. Any alteration renders that thing invalid. And so that nobody will steal your manual, you come back and say that somebody has stolen your manual. You're on your own. No? So please, think I'll be able to tell you more on Saturday by God's grace. Okay, guys, do have a wonderful day and stay out of trouble. See you in lab on Monday. Some people are still dropping attendance. How do we get the manual? The manual is sold at the bookshop, please. The manual is sold at the bookshop. Ask your colleagues. Sir, how do we get the slides? You will get a recording of the lectures, which is more important than the slides. Okay, do have a wonderful day. Bye.